Hello, this is Ed Chapman, and this video cast is going to explore some of the in vitro tools that are used by biotechnologists to amplify and analyze DNA samples. The list of tools that we're going to focus on include polymerase chain reaction, or PCR, gel electrophoresis, RIFLIP analysis, southern blotting and DNA probes, dideoxy chain termination sequencing, and RNA interference, or RNAi. This video cast is going to focus exclusively on PCR. All right, PCR stands for polymerase chain reaction, and it's a relatively simple way to amplify or make lots of copies that are identical of a limited DNA supply or a sample that contains a very small amount of DNA. It's pretty simple in the way it works. It's based on a heating and cooling cycle accomplished by a machine called a cycler. An enzyme called DNA polymerase, which of course is the enzyme that builds DNA. In this case, it's a DNA polymerase enzyme that can withstand temperature changes. A supply of nucleotides, which remember are the monomers needed to build DNA. And of course, some primers, which tell DNA polymerase where to start building. The primers in this case are made of DNA, not RNA. What has to happen is the primers that you pick for this process have to bracket or surround the gene that you're interested in sequencing on both ends. And you'll see what that means when we start the video. Sorry about the ad.
The polymerase chain reaction is a method for making many copies of a specific segment of DNA, starting with a very small amount. This technique can be used to identify specific microorganisms from a small amount of DNA, and to identify persons involved in crimes from DNA on cigarettes or in a single hair follicle. The DNA to be amplified is mixed with deoxyribonucleotides, a thermal stable DNA polymerase called Taq polymerase, and DNA primers. The DNA primers hybridize to the ends of the gene to be amplified and provide a starting point for the Taq polymerase. The mixture is heated to break the hydrogen bonds in the DNA, forming single-stranded molecules. The mixture is then cooled sufficiently to allow the DNA primers to anneal to each end of the segment to be copied.
Okay, I'm going to pause it there. Notice that these primers have attached themselves to the code at both ends of the gene of interest. There's a wide variety of primers to choose from in this technique, and you kind of have to play around and find the right ones that actually bracket or are close to the beginning and the end of the gene of interest that you wish to amplify.
Taq polymerase then synthesizes the complementary strand of DNA using the primer as the starting point. The temperature is raised again to separate the DNA strands and then lowered sufficiently to allow the primers to attach. Taq polymerase now synthesizes another set of new complementary strands. This process is repeated until enough DNA has been produced to be identified or used for further research. After 21 cycles, one molecule of DNA can be amplified to over 1 million copies.
Okay, notice that to make this process work, all you have to do is get it set up, and then the machine takes over and just heats it and cools it and heats it and cools it and heats it and cools it over and over again until you get the copies, the number of copies that you need. And as I said, this is a relatively simple process.
The amount of amplification can be achieved by running the reaction overnight in a thermal cycler, an instrument that automatically raises and lowers the temperature at appropriate time intervals.
All right, we'll stop there. Thank you, Mr. Video Guy. Go back to our PowerPoint. All right, and why would you need to use polymerase chain reaction? Well, sometimes evidence that's collected at crime scenes exists in only a really small amount, like a single little drop of blood on the sidewalk. That's kind of a famous example from the O.J. Simpson trial. Or sometimes you have you're doing research in a lab and you need to share the DNA that you've collected with other people. Okay, you have a small amount, so you want to make more. PCR is a way that you can expand your DNA, cloning it millions of times so that you can share it with other people. And lastly, you may have a market. There's a market out there for certain genes, and you may want to, sell it to different people. So people definitely amplify DNA for sale. Thanks for watching, and that's the end of this video cast.